Since we are omitting some of the details from Chapter 11, all of Chapter 11 will be in one video. We still have, though, three major ideas we want to cover in Chapter 11. We want to look at how gene expression is controlled. We want to take a brief look at cloning and then the genetic basis of cancer. So this first part takes a look at the control of gene expression. What this really looks at is how do different cells in your body turn on and off particular genes so that we can make the right proteins for that particular cell. So cells that are in your small intestine will be likely producing much different proteins, certain digestive enzymes, than cells that are in your skin. Different cells, depending on where they are, have to express different genes, but they all have the same basic code. They all have the same set of blueprints. So how do they do that? How do we go from a cell that has all of these instructions to one that is only expressing certain genes? And when we say they're expressing certain genes, it means that those genes are being turned on and the products of those genes are being made. So first we'll take a brief look at prokaryotic gene expression, because gene expression is quite different in prokaryotic cells versus eukaryotic cells. It's quite a bit more complicated in eukaryotic cells. Keep in mind, prokaryotes are all single-celled organisms, so they don't have the division of labor like we see in a multicellular organism where different cells perform very different functions. But overall, the process where we decide in a particular cell at a particular time what genes we're going to use and what proteins we're going to make, that's gene expression. One way to alter gene expression is to turn on and off certain genes. Now, one other way we can think about it is not only can we turn them on and off like a light switch, but we can also speed them up and slow them down, kind of like a dimmer switch. So there's a couple of different ways that we can do this. In prokaryotic cells, genes are organized in units that typically are responsible for controlling related mechanisms. For instance, if you're getting ready to break down a particular sugar, let's just say that we need to get the sugar inside the cell, and there's a protein that does that. And then let's say we need to break this disaccharide into two monosaccharides, and there's an enzyme for that. And then we need to do something else with these monosaccharides. So let's say we need 3 different enzymes to effectively do this. What we often find is that those 3 enzymes that are required to perform that one function will be located next to one another in our genome, so that when we when we flip that light switch on for one gene, it's on for all 3. Because it wouldn't make a whole lot of sense to turn on one of those and not the others, because we need all 3 together. And that is the concept that you see here of the operon. So an operon is this unit that we control as a single unit, but it will have several genes on it. So when we turn on the operon, we can essentially transcribe each of those genes together. We also have proteins that will bind to different control sequences in the DNA. There can be on this DNA a particular area where a regulatory protein can come in and bind to allow us to get access to those genes, to turn on those genes or to turn off those genes. So we have a lot of these different regulatory proteins that get involved as well. This is an operon, which means it consists of multiple genes. So this operon consists of these 3 genes right here. All 3 of these genes are important for the cell's ability to break down lactose and to use lactose as an energy source. Lactose is a type of sugar. Some of the major pieces of an operon include a promoter, an operator, and then we have the genes here that are actually going to be transcribed. All of this, the promoter, the operator, and these 3 genes, which we sometimes call structural genes, they're called structural genes because they actually make a protein. These are called regulatory sections because those sequences don't get converted. We don't actually transcribe anything off of these areas. These are simply giving us access to these 3 genes over here. In order for us to transcribe a gene, if you remember from our previous section, RNA polymerase has to come and bind to this promoter. That's what initiates this transcription process. We have to get the promoter and the polymerase to bind to one another. There's a section between our structural genes and the promoter that's called the operator. And the operator will essentially either block or not block the ability of this polymerase to bind. So right now there's a protein sitting there, and that protein is making it so this RNA polymerase cannot get into the promoter. So this protein right here is acting as what we call a repressor, because it's repressing transcription. It's not allowing transcription to occur. So this is essentially turning it off. The repressor has turned it off. We can't bind the RNA polymerase to the promoter. Polymerase must bind to the promoter for transcription to begin, and polymerase will move down there. With this protein attached here, this gene is turned off. Now there is a binding site you can see right here on this repressor protein, that if another molecule binds there, it can pull this repressor protein off of the operator. And that's what we see going on down here in the bottom section. This lactose molecule has bound to the repressor has moved off of the operator, and now polymerase comes in, binds with the promoter, and transcription occurs. So up here in the top, we are in the off position. It cannot transcribe. On the bottom, we're in the on position. It can transcribe. This lac operon is what we refer to as an inducible operon. It is normally in the off position unless a molecule induces it to begin. So it's off unless we turn it on. The molecule that is turning it on is lactose. Think about what's happening. We need to transcribe these 3 genes to break down lactose. If lactose isn't there, the gene is turned off. When lactose is present and we need to break it down, lactose removes that repressor protein. Therefore, we can transcribe the genes, we can break the lactose down. Now think about what's going to happen in the next step. We're transcribing this gene, we're breaking lactose down. What's happening to lactose? It's declining in the cell, right? Because we're getting rid of it. And we're going to get rid of it to a point where now there's no lactose. When that occurs, this will turn off again. So this is turning on when we need it, and it's off when we don't. This compares that lac operon, which we described as inducible, off until you turn it on, to another operon called the tryptophan operon, which is what we call a repressible system. This repressible operon is normally in the on position unless we turn it off. Lactose operon is off unless we turn it on. That's inducible. The tryptophan operon is repressible. It is on until we turn it off. The interesting thing about the tryptophan operon is this is not something we need to break down. This is something the cell needs to build. This is an important amino acid that we need in cells to build proteins. If we don't have enough tryptophan, we need to build tryptophan, and there's enzymes that we have to use to do that. Normally this is on and we're making tryptophan. If the tryptophan level gets high enough where we don't need any more tryptophan, that tryptophan comes and turns this off. It makes it so that this repressor can bind to the operator and therefore stops transcription. So this is something the cell is making. If we get too much of it, we turn it off. This is something the cell needs to break down. If it's present, we turn it on. Let's take a look at differentiation and gene expression. Differentiation is the process of specialization in a cell, where that cell becomes specialized for a particular function. So you could imagine where we all started off as a fertilized egg. That cell has the ability to become any type of cell in your body. At some point, though, there's a certain cluster of cells that are destined to become neurons and nerve cells, and there's other clusters of cells that will become cartilage and bone. And at some point, those particular cells can't go the other direction. They can't revert back to becoming from a cartilage cell back to a nerve cell. So they undergo this differentiation where they start to specialize in their structure and function. So the differentiation is this acquiring of the specialized functions for that particular type of cell. And of course, in multicellular organisms, we undergo lots of differentiation because we have lots of different cell types and tissue types that have to perform certain functions. Each one of those cells that has differentiated to become a particular type of cell has certain genes turned on and other genes turned off. And it's this change in gene expression that allows cells to differentiate, to become cartilage cells or bone cells. It's not the genes that are present, because remember, all of those cells in your body have exactly the same set of blueprints. They have all the same genes. It's not what genes are present. It's what genes are on and what genes are off, the timing of those genes as they turn on and off. That's what makes one cell function differently from another cell. There's a lot of different ways that this change in gene expression can occur. We just looked at the possibility of turning operons on and off by using certain proteins in the cell. And let's look at some other types of things that can alter this gene expression. Part of this gene expression can be regulated by looking at how chromosomes are packaged. Remember, chromosomes are packaged where the DNA molecule is wound around these proteins called histones. Each one of these is a histone. You've got a group of histones that we've wound this DNA around. And then one of these little packages with the histones plus the DNA around it is called a nucleosome. And then we further package that. So we saw before where we loop it around and we package it in lots of different ways. This structure can influence how genes get turned on and off. This packaging can block gene expression in some areas and allow gene expression in other areas. The best way to probably think about this is to imagine how tightly packed is the DNA in a particular area. If it's very, very tightly packed, it may be that that area of the chromosome is not really accessible. There might be other areas where it's more loosely wound that it is accessible. And all the different cells will have different areas that are more tightly packaged than others. Just the overall chromosome structure allows us differential access to different regions along these chromosomes. One example of how DNA packaging influences gene expression is this phenomenon known as X chromosome inactivation. This occurs in female mammals. If you go back to our genetics chapter, remember we have some genes on the X chromosome. They're called X-linked genes. Now females get 2 copies because we have 2 X's. Males only get 1 copy of X because their other chromosome is a Y. So any genes that are sitting on X, males only have 1 copy. Females have 2. The way that the female cells deal with this extra copy is they turn one off. In any cell, one of those X chromosomes is inactivated. A good example of showing X chromosome inactivation is in calico cats. Typically, we don't see male calico cats. And if we do, there's probably another explanation for it. What happens in a calico cat is there is a gene on the X chromosome for orange fur and black fur. And this calico cat gets 1 copy of each. It's a female. It has 2 X's. On one of its X chromosomes, it has the orange allele, and then on the other one, it has black. During embryological development, one of those is going to get turned off. If we inactivate the one with the black allele, we see a splotch of orange fur, where those cells then divided. We have the cell that inactivates the black, it expresses the orange, and there's a clump of cells there that came from that one cell that has the black one inactivated. And then we have a clump of black fur where the orange one has been inactivated. Once again, we don't see this in males because males only have 1 X chromosome. In the rare cases where we do see a calico male, it could be an XXY. Remember, non-disjunction sometimes can give you a cell that has an extra chromosome. So if a male had XXY, that would possibly explain where you would get a male that has this kind of a pattern. But it also explains why it's way more common in females and very rare in males. Another thing that can influence our expression of genes is what we call epigenetic inheritance. Epigenetic literally means on top of genetics. What happens is there's a change that occurs along the chromosome, but it's not a DNA change. So it's not a change in the base pairs. It's not any change to an A, G, C, or T along the DNA strand. It is a change to molecules that associate with the chromosomes. Some of these are the histone proteins, so we can have a change in our histone proteins. We also can have a change in what we call these tags, which are actually methyl groups. So we refer to this as methylation. We can add methyl groups along the strand of the DNA molecule. Physical environmental factors can cause this to happen. So certain things we are exposed to throughout our lives can put these tags on our DNA. And that can change the way those cells express genes. So it could take a normal cell and create a cancer cell, and it could cause aging and lots of other different things. Epigenetic inheritance is something that's relatively new. Probably within the last 10 to 15 years, they've really started looking at it. We still don't know a lot about it, but we do know that it happens. We do know that it changes gene expression. And the interesting thing is, years ago, they thought that all those epigenetic marks that we pick up through our life got stripped away when we made egg and sperm, and so didn't go to the next generation. And now they're finding that that's probably not true, that some of them do go to the next generation. In addition to proteins and the packaging of chromosomes and the epigenetic factors, we can also control transcription in eukaryotic cells, kind of like we did in prokaryotic cells with the lac operon and the regulatory proteins. It's just a lot more complicated and complex when we look at it in a eukaryotic cell. So we do turn genes on and off, but we have a lot more complex ways of doing it. There's a lot of different regulatory proteins that interact with the DNA that can turn the transcription of eukaryotic genes on or off. We also can increase the speed at which we transcribe different genes. So we can turn them on and off, and we can speed them up and slow them down. In general, these proteins that we look at that control transcription are referred to as transcription factors. There's a lot of different types of transcription factors, and we won't worry about each of those individual ones. But these are essentially proteins that help the binding of RNA polymerase to that promoter that has to occur for transcription. So if we can take a protein that will help bring those together and help polymerase bind to the promoter, we can facilitate that transcription occurring more efficiently and faster. Here we'll see an animation that looks at transcription and the initiation of transcription.
Most of the fine-tuning of gene expression occurs at the transcription level, making an RNA copy of a DNA sequence. Recall that to initiate transcription, an RNA polymerase must bind to the promoter region adjacent to a gene. This cannot occur without the help of specific protein transcription factors. Some transcription factors, called activators, bind to enhancer regions some distance from the gene. The DNA bends, bringing the activators closer to the gene, where the activators interact with other transcription factors and RNA polymerase to initiate transcription.
So in addition to looking at genes being regulated by altering whether or not transcription turns on or off or the speed of transcription, we also know that in eukaryotic cells, after you make your messenger RNA, we edit that messenger RNA. Let's take a look at how we might alter gene expression through this splicing mechanism. Remember, when we have our section of DNA, some of the sections in here are express sequences. We will actually turn them into a protein. And other sections in here are not expressed. Those are the introns. So here they've got the exons labeled, 1, 2, 3, 4, 5, and then these light sections in between are the introns. Those are the ones that we're going to pull out. And then we're going to splice back together those exons. If I splice my messenger RNA in different ways, I actually could get a different product. There's nothing that says that we have to splice them back together in exactly the same order they were. We could splice them together in a different order. And if we do, from the same gene and the same exons, we could actually get a different messenger RNA, which would give us a different protein. Another way that we can have different cells doing different things is by altering the way that we splice and edit the messenger RNA that we just produced. Same transcript, but a different edited transcript and a different protein. In humans, about 90% of our protein coding genes undergo alternate splicing. So 90% of those have alternate ways that they can be spliced and get different proteins. So here we'll take a look at RNA processing.
A cell can rapidly adjust to environmental changes by regulating several processes that happen after transcription. In RNA processing, A cap and tail are added, introns removed, and the remaining exons spliced together. Alternative splicing of RNA may create different mRNA molecules from the same primary RNA transcript.
Now, after we process the messenger RNA, we still have the ability to come in and regulate gene expression because there's a whole lot of other things that happen to proteins before they result in a phenotypic consequence. One of the things we can vary is how much of the messenger RNA molecule is in the cell, because how much messenger RNA is sitting there can determine how much protein we make. That, in addition to some other factors that influence translation rate, can influence how that messenger RNA is used to make proteins. So if I have a lot of this messenger RNA sitting there, I might be making that protein very rapidly. And if I only have a little bit of messenger RNA, I might make it slowly. One of the things that we can alter then is how quickly we allow the messenger RNA to be inactivated. These messenger RNAs are fairly short-lived. They don't stick around forever. We make them, we use them, and then we typically break them down. However, in some cases, those messenger RNAs are a little bit longer-lived, and in other cases, they're shorter-lived. Different cells might allow the messenger RNA molecule to live for a longer period, and that would allow it to produce more protein. That then could make one cell behave very differently from another cell. We also might go back and look at some of those non-coding sections, those non-coding RNAs. Only about 1.5% of the human genome actually codes for proteins. And that's true for lots of multicellular eukaryotes. It's not just the human genome. What is the 98.5% of the rest of our genome doing? We don't know for sure. We are looking into that, and sometimes that area is called the dark matter of DNA because we're not sure exactly what it's doing. But it's not coding for particular proteins. The idea is that those non-coding RNAs and those non-coding sections of DNA might also be functioning in gene expression and the control of gene expression. Some of the DNA has genes for making ribosomal RNA and transfer RNA. We looked at making of messenger RNA, but we also saw the role of transfer RNA in bringing in the amino acids when we're building a protein. Well, that has to be made somewhere too, and we make that back off of DNA as well. Same thing with the ribosomes. Remember, ribosomes were made up of ribosomal RNA and protein. Where do we get that RNA? The instructions are back in the DNA. One other way that we could influence the making of proteins in a cell is by altering the production of our transfer RNAs and the production of our ribosomal RNA. A lot of recent evidence suggests that a fairly large portion of our genome is transcribed into functional RNAs, but not protein coding RNAs. So they can have some function, they're just not there to make traditional proteins. Probably one of the best ways to think about gene expression and how different cells do different things is to try to imagine all the steps along the pathway that a cell goes through when we go from DNA all the way to protein and then the expression of some phenotype. The production of insulin, let's say, for a particular cell. What are all the steps that we have to go through in a eukaryotic cell to make that happen? Because every one of those steps is a potential for us to control it and to regulate it. So let's go through those steps. If we go back and we look at the chromosomes, we already saw that we have potential for regulating which genes get expressed because of the packaging of those chromosomes. The histones, the looping and coiling of the DNA, clearly we can control that and we can regulate that. So we can give access to certain parts of the chromosome and not access to other parts of the chromosome. We also know that we can alter transcription. We have these transcription factors that can increase the rate of transcription. We have others that can decrease the rate of transcription. We can turn genes on and we can turn genes off. Then we also know that we're going to take that particular transcript and we're going to edit it. By splicing it in different ways, we can get different products. So we can alter gene expression by alternative splicing mechanisms. Now this messenger RNA is going to go out into the cytoplasm. This is actually another potential for control because we can control how quickly this messenger RNA moves from the nucleus out into the cytoplasm, and we can control where it goes. So we have potential for control there as well. We also said that messenger RNA doesn't live forever, and we can control how quickly we break it down. So we can make a lot of a protein if we don't break it down quickly, and only a little bit of a protein if we break it down very rapidly. We also then can control it at the next step, at the step of translation. Right now, we just have a messenger RNA. We haven't done anything with it yet. Now we can control how quickly we turn that RNA into a protein. So we can control the rate of protein synthesis. When we do translation and we make this polypeptide chain, remember, not all proteins are a single polypeptide chain. Some of these proteins are multiple chains, and many of these proteins require the ability to be folded properly in order to function a certain way. And sometimes you can take a particular polypeptide chain and fold it differently. If I fold it one way, I might get a particular protein, and I could take this polypeptide and fold it a different way and get a different protein. Then I can also modify that protein. So remember, when we were looking at cell membranes, we looked at glycoproteins, where we attached carbohydrates to proteins, or lipoproteins, where we attached lipids to them. Or if we look at hemoglobin, we have a protein, but we have iron in the middle of it. So we can go in and modify these proteins. That can be controlled and regulated as well. Activating these proteins. So sometimes proteins are in an inactive form, and it takes a particular either ion or some process to activate it. So we can control the activation of the proteins. Just like our messenger RNA, not all proteins are going to live forever in a cell. We will break them down after a while. We can regulate how fast those proteins get broken down. How long will that protein perform its function in that cell? That can vary as well. So we can regulate it at that step. If we look here, we see that we have many, many potentials along this pathway for cells to regulate what proteins are being made, how much of it's being made, how fast it's being broken down. All of that allows that cell to express a different function from some other cell that it's doing this a little differently. Here's an animation that shows us a blocking of translation. This animation shows us a little bit about this messenger RNA degradation.
Translation of specific mRNAs may also be blocked by regulatory proteins that bind to sequences within the untranslated region at the 5' end of the mRNA molecule, modulating the amount of protein produced.
This animation shows us protein degradation.
A cell can get rid of abnormal or damaged proteins and limit the lifetime of functional proteins by means of selective degradation. The cell marks the protein for destruction by attaching small ubiquitin proteins. Giant protein complexes called proteasomes recognize ubiquitin and break down the tagged protein.
Here we'll see an animation of protein processing.
Often, polypeptides must be modified in some way to produce functional proteins. For example, a polypeptide is cleaved in 2 or more polypeptide chains. Other molecules might be attached to the protein, or the protein might be phosphorylated. Some proteins need to be transported to specific destinations.
So now that we've talked a little bit about how different cells can manage to express different genes, let's ask questions about how they get the information, the signal, to tell them what genes need to be expressed. So we saw all this happening inside the cell, but what is it that the cell is listening to get this information to say, well, I need to make insulin now, or I need to make a neurotransmitter now? What information is coming into that cell to tell it that? And this brings us to cell signaling. We see a lot of different RNAs and proteins that are produced that control development of a cell from fertilized egg to an embryo. These RNAs and proteins must be telling different cells within that developing embryo what to do. We have particular genes called homeotic genes, or sometimes you'll see these referred to as Hox genes, H-O-X. Hox genes are master control genes that regulate groups of other genes. And these genes are very important in determining the basic body plan of an organism. Where does the head go? Where does the tail go? Where do we put the limbs? What's the the top side? What's the bottom side of a developing embryo? The really interesting thing about Hox genes is that they are highly conserved in evolutionary history. So some of the same Hox genes that we would find in a fruit fly, we also find in vertebrates. The ones that determine where the wings go in a fruit fly are basically very similar Hox genes to what we have that tells where limbs go in a vertebrate body. So they were something that evolved very early on in multicellular organisms. So these are genes that boss other genes around. These other genes are sometimes referred to as switches. And so we have switches that turn on genes, and then we have Hox genes that tell switches what to do. Here we'll see an animation about cell signaling. How does the information get into the cell to tell that cell what genes to turn on and off?
Cell-to-cell signaling, with molecules carrying signals from sending cells to receiving or target cells, is a key mechanism in the coordination of cellular activities. In most cases, a signal molecule acts by binding to a receptor protein in the plasma membrane of the target cell and activating a series of changes, a signal transduction pathway, that triggers a specific response within the cell. A signal molecule binds to a specific receptor in the plasma membrane of the target cell. This activates a series of relay proteins, which in turn activates a transcription factor. The activated transcription factor attaches to the DNA and triggers transcription of a specific gene. The mRNA message is translated into a new protein, which changes the structure or function of the receiving cell.
How do we actually go in to a developing embryo or individual who maybe has some disease within particular tissue and figure out what's going on at the gene expression level? There's a variety of techniques that we can use to show and to study how genes turn on and off and to look at what genes are functional in certain tissues. One of the techniques is nucleic acid hybridization. This allows researchers to go in and identify particular cells where a target gene is being expressed. So if I want to ask in what cells is this insulin gene being expressed, I can use nucleic acid hybridization to identify those cells that are expressing it. We also have something called a DNA microarray. The microarray has been huge in biotechnology because it allows us to ask within a tissue what genes are on and what genes are off. We can screen for lots and lots of cells and lots of genes at a particular time. So we can come through here and we can look at certain genes are on in particular cells, other genes are on in other cells. And we can actually almost look at this kind of like a visual symphony. We will see at different points in time, different genes coming on and going off in different cells. And the amount of that gene that's being expressed in different cells is changing over time. What is it that then signals cells to transcribe, translate, and regulate the production of certain kinds of molecules? Think of a developing embryo. How do two cells that are growing towards one another, because that's how a lot of this works, is you'll have cells that are actually growing towards one another, and then they'll make a connection. How do those cells communicate with one another just to stop dividing once they hit each other and to guide one another? So they send out these signals that will guide one another to come towards them so that they can actually interconnect. Cells send out these signals that coordinate the activity of other cells. In most cases, we have a signal molecule that will go and bind to a receptor protein on the plasma membrane of the other cell. So in number 1, we see that the cell is sending a signal. It's going over, and in number 2, we see that that signal is binding to a receptor protein on this other cell. In number 3 is what we call signal transduction. This is just a way of saying that this signal that came in is going to be conveyed throughout the cell until it gets over to number 4 here, where we're go into the nucleus and decide what sections we're going to activate. So remember, in eukaryotic cells, we have these transcription factors that will come in and activate certain genes, allow the promoter and the polymerase to bind to one another. This also means that a signaling cell on this side will only communicate with certain types of cells. They're not going to communicate with all types of cells. Other cells that they don't talk to don't have the right receptor proteins there. The sender and receiver are very specific. Here's an overview of cell signaling. Next, we want to take a brief look at cloning in plants and animals. So a clone is an individual organism created by asexual reproduction, and it will be genetically identical to that single parent. Cells within an individual's body that are capable of becoming any type of cell in the body are referred to as totipotent. So you've probably heard of embryonic stem cells. Certain stem cells have the ability to become any type of cell in the body, and those are totipotent cells. Regeneration is regrowth of lost body parts, and the reason they bring this up here is because regeneration demonstrates that even though certain cells have become differentiated, does not necessarily mean that that limits then that cell's future genetic potential. Over here we see the root of a carrot plant. They can take cells off of this carrot, place them in a culture growth medium, take one of those cells, put it on a culture dish, get a small plant out of that, and then grow into an adult plant. That means that those cells that were here in the plant have the ability to become any part of that plant. So those cells were totipotent. They were able to become any type of cell. One way to do animal cloning is called nuclear transplantation. What we do here is we take DNA from a donor cell and insert it into a host egg that does not have a nucleus in it. So you're essentially using the host egg just for the cytoplasm and putting a nucleus in that. So the resulting is a clone of whatever the DNA donor was. When you take a fertilized egg and it divides many times, you end up with sort of this ball of cells. And that's what this blastocyst is. It's a very early embryonic stage. So in a mammal, of course, we have to get that embryo to implant into a uterus. So what they'll do in a mammal, and they call this reproductive cloning, is they will remove the nucleus of the egg. They will take a donor cell, and it can be a somatic cell from the donor, inject that into that cell. So now we have a nucleus that's been added to this egg, allow it to grow up into this blastocyst, and then that blastocyst will be implanted in a surrogate mom, and then the offspring that is born from that is actually a clone of whatever DNA we had in this donor cell. There's another type of cloning that's called therapeutic cloning, as opposed to the reproductive cloning. The goal here is to produce embryonic stem cells. So these cells, because they have the ability to become all sorts of other tissue within the body, can be used therapeutically. We can use them to replace bone marrow, for instance, if a person has bone cancer. If we use embryonic stem cells, we would take embryonic stem cells, remove some of them from this blastocyst, culture them. We could put them in under different culture conditions, which would then cause the formation of maybe heart cells, nerve cells, blood cells. We also can get some types of stem cells from adult tissue. We can't get all types of stem cells from adult tissue, but we can get certain types. So we can get some stem cells in bone marrow that can then be used to produce blood cells, because this is where the blood cells are produced anyway. So we can use stem cells from here in the bone marrow to create blood cells to treat something like leukemia. But the adult stem cells are limited because we only have certain types of stem cells in the adults. So yes, we have cells that will give rise to all the different types of blood cells that are located in bone marrow, but we don't have stem cells for all of the types of tissues that we have in adults. Now let's take a look at, briefly, the genetic basis of cancer. Cancer results when we have uncontrollable division, which means there's a mutation somewhere in a protein that controls the cell cycle. So a mutation can change what we call a proto-oncogene. This is a normal gene that controls cell division or helps control cell division. But if that gene has a mutation, it can be converted into an oncogene, which causes that cell to divide excessively. And that then can lead to the formation of a tumor because we have uncontrolled cell growth. At the bottom, we see a diagram of this. We have this proto-oncogene. This is a gene that is used to stimulate cell division. Here we have a mutation within that gene, and this mutation causes a hyperactive growth. So we have too much of this protein being produced and excessive growth. So it is now an oncogene. Mutations can also turn off certain genes that are known as tumor suppressor genes, and that can also lead to cancer. So tumor suppressor genes are genes that prevent this uncontrolled division. And so if we have this tumor suppressor gene that is functioning correctly, it will produce a growth inhibiting protein which will keep cell division under control. If we mutate the tumor suppressor gene, we can have a defective protein here, and so we no longer properly inhibit the growth, and we get uncontrolled cell division and the formation of a tumor. Many different changes can underlie the development of cancer. So it can be a whole series of genetic changes. Colon cancer illustrates that gradual progression from somatic mutation to cancer pretty well. So here we have DNA changes going on and cellular changes that they're tracking. So at the DNA level, we see the oncogene is activated. That causes an increase in cell division. We also see a change in DNA that can suppress that tumor suppressor gene, inactivate that, and we see the growth of a polyp. And then a second tumor suppressor gene could be inactivated that would allow the growth of what we call a malignant tumor. So here we have a polyp, which is a benign tumor, and then a second mutation results in the growth of a malignant tumor. This figure on the bottom is showing us that sometimes it's the accumulation of these mutations that gives us this malignant type of cell. So we see a normal cell, and then here we have one mutation in one area of a chromosome. In the second one here, we have two mutations, and we start developing these mutations. And as we add mutations in, we're more likely to develop one of these malignant cells. So it can be the sum of many mutations that causes the formation of this malignant tumor. Another thing that can happen is the signal transduction pathway that we looked at before that allows us to communicate to cells that will then go in and turn genes on. That transduction pathway can be broken. And if we have a faulty transduction pathway in a cell, that can drastically influence the cell cycle. Some of that control of the cell cycle is through these signal transduction pathways. So if we have an error in the transduction pathway, we can certainly interfere with the normal cell cycle. So many of our proto-oncogenes and tumor suppressor genes interfere with this signal transduction pathway. We don't need to worry about the details here, but that's one of the things they're showing here. We have a normal cell that is responding to growth factor, and then we have the signal transduction that goes through here, and we get normal cell division. Over here on the right, we have a mutant cell, and so this mutant cell, there's no growth factor that is being produced, so we don't get the normal product of this particular pathway. We don't get the proper control. And so what happens down here is we overexpress one of the proteins and causes increased cell division. We have another example here where we have a growth inhibiting factor, and we have the normal pathway here. So this is going to be something that inhibits growth. Normal pathway here, we get no cell division. So this is a pathway that would inhibit cell division. Here we have a mutant cell, and this transduction pathway is not working correctly. And so here, instead of us receiving the signal for the cell not to divide, it increases cell division. So in both cases, we had cell division where we should not be getting cell division. We also know that with cancer, there's a lot of external factors. many lifestyle choices that we make, depending on types of food that we eat, exercise, many other things can influence whether or not these pathways are activated. Most cancers arise from a mutation that is caused by some sort of environmental factor, and we call those things that are in the environment that can cause cancer, we call them carcinogens. So obviously we would try to reduce our exposure to carcinogens to reduce our risk of developing cancer. We're at the end of chapter 11. Make sure you can go through and make sure that you can answer these questions.